morning. How are you?
Hi, can you hear me?
Yeah, I can hear you. I can hear you
Okay,
fine.
great.
So I can try to put your slides up, make sure that's working. I can see you and hear you.
Okay, great. So let me just try to do that.
Well, thank you for agreeing to present on such short notice.
Oh, well, thanks for inviting me. I mean, I know Jake very well, so I mean, we go all the way back. So
Oh, did you
I
know him
mean,
in China?
thanks for thinking about me. So
Did you know him in China?
yes, so we actually are classmates at Peking University, and then I mean, we certainly did not know that that many years after we still sort of, I guess, like colleagues and do share similar research interests. But it was a fun ride. Yeah, I mean,
Wow.
I know him a long time. I mean, this was
Wow. I'm sorry he's
back
in
in
Switzerland.
1990. So that's like what
Wow.
is
Well,
it,
sorry he's in Switzerland. So
30 plus. Say it
he
again.
may or may not be able to join because
No
he's
worries, don't worry. I just saw him. I saw him in the ISMV
Oh, okay.
in the summer. So yeah, we had dinner together. So it wasn't that long ago. So let me try to share my screen. Share screen. How do I show? Oh, here. Yeah.
Oh, looks perfect.
Great. Awesome.
I'm gonna turn on. I'm going to turn off myself because you're the star. So, and people always show up at the last minute and late.
Yeah, no worries. Yeah.
And go ahead, Jake.
Okay. Well, welcome everyone to the Bioinformatics Power Talk series. We have this series for over 6 years now. And we're very happy to have Professor Brendan Xia to give us a webinar. I know Professor Brendan Xia probably for more than 27 years, primarily because he's one of the rare people I would consider to be a prodigy. And I really, literally, I mean it. If many of you know about the Science Olympic, when he was a high school student, he represented China to one of the gold medal in Olympic. I think it was in chemistry, if I'm not mistaken. So that means that at least for that year, he represented the best of the best of high school. And so he went to Peking University, but shortly after, he took the chemistry PhD program training at Stanford. After Stanford, he was trained with Professor Mark Gerstein, who some of you may have known that in 2019, we invited Professor Mark Gerstein as a keynote speaker in the MC Bios annual meeting. So Professor Mark Gerstein at the time was very interested in protein interactions and network biology. And I believe that is sort of the turning point, and I'll let him speak to it. And then he switched from chemistry to computational systems biology. And since then, he has published many key papers and important work in the field, and took a faculty position at Boston University before he took the Canadian Research Chair professor and one of the founding members of the Biomedical Engineering Department at McGill. And so he's certainly more qualified than me to talk about the systems biology. And without further ado, let's welcome Professor Xia.
Thank you very much, Jake, for the really, too nice an introduction. I just want to say that I want to thank Amy and Jake for inviting me. And like Jake said, we have known each other for a long time, and I have followed Jake's work for many years, and we share a lot of interests in systems biology, network biology, protein modeling. So here at McGill, unfortunately, we do not have an centralized informatics institute. Instead, the biomedical informaticians like myself are scattered around in different schools and different departments. So in my case, I am in the bioengineering department in the School of Engineering. So I want to talk a little bit about how I justify my existence. But I think it's it's sort of a good phenomena that you find biomedical informaticians everywhere. They play very important roles in different parts of the university. So I would like to talk about multiscale modeling of biomolecular networks. And I don't think that this audience needs an introduction regarding the importance of network biology and systems biology today. There has been a lot of excitement. People are working towards producing a map of all kinds of biomolecular networks. For example, in human, this is an example of a human protein interactome. Here, each node represents a human protein, and each edge here represents a physical interaction between two proteins. But this is only one of the many kinds of networks one can study. Many other kinds of regulatory networks, metabolic networks, you can all study by these kinds of large-scale combined experimental and computational methods. So where I come into play is I had a training in structural protein modeling. So the idea is that these kinds of network maps, they are very exciting, they're very important, but still they represent a kind of a high level, right? A high level view abstract view of what's going on inside the cell. Each one of these nodes is not just a dot, but rather is actually a three-dimensional structure of a protein machine. And each one of these edges, it represents a precise interaction between two proteins that's mediated by a specific set of residues called interfacial residues. So I'm very interested in bridging the gaps between network biology and structural biology. So the idea is to be able to model each one of these protein nodes in terms of a three-dimensional structural model, and each one of these edges, protein-protein interaction edges, in terms of a three-dimensional structural model of protein complexes. So why is this important? So this goes back to how I justify my existence in the engineering department, is because a lot of people my colleagues are interested in molecular cell engineering. But my argument is that in order to do, in particular, rational design, rational engineering, you've got to know how your system works. And I think it's super important to produce some kind of a model, a working model of, let's say, a cell. It doesn't have to be extremely detailed, but it needs to have some predictive value, right? And in order for a model of the cell to have some predictive value, it needs to connect the causes with the consequences, right? Now, if you think about disease, in particular human disease or development, a lot of these causes happen at atomic scales. You got a point mutation, which causes a change in the amino acid or in the genome, but then it gets cascade, it gets blown up out of proportion and lead to network level, cell level consequences. Cells are not happy, and you got disease. So what this means is the model of the cell needs to have at least two levels, right? The level of the cause, which happens at the atomic level, and the level of the consequence, which happen at the level of the networks and beyond. So that is how I think about it. So that's why we decided to focus on these two, at least these two different levels, and produce a model of the cellular networks, but with these kinds of atomic details. So the way we do structural modeling for these, so we start with an experimentally determined protein-protein interaction network. So we don't predict protein-protein interactions. We leave it to the experimentalists to detect protein-protein interactions. So what we do is we take experimentally confirmed protein-protein interactions and try to build structural models for them. So how do we do that? We use an established technique called homology modeling, also known as template-based modeling, to produce these structural models. So this is how it works. Let's say we have a protein. Here's a protein of interest. We know its sequence, but we don't know its molecular structure. So what we do is we do a sequence similarity search of this query sequence against the PDB, the protein data bank, a library of proteins with solved three-dimensional structures. Now, half of the time, well, actually more than half of the time, we'll get lucky. We'll find a significant hit in terms of sequence similarity. And we know that if there's a high level of sequence similarity, then most likely structural-wise, the two proteins will share very similar structures. So we can then borrow that structure, that protein, that different protein with a solved structure, and use it as a template to build a structural model for my query protein. This is called homology modeling for a single protein. Now you can do the same thing for protein-protein interactions. So now we have a pair of proteins that are known to interact experimentally. Again, we're not trying to predict new protein-protein interactions. We're taking known protein-protein interactions and we're trying to add structural details, atomic structural details to that interaction. So what we do is we search this pair of protein sequences against this library of protein pairs, these other protein pairs whose structures for their complexes has been solved by other experimentalists. So in this case, sometimes we'll get lucky. We will find a significant hit in terms of sequence similarity between my query sequence pair with, let's say, this particular protein pair, protein complex in the PDB. In that case, we can then assume that my new query protein sequence pair will adopt a similar structure as that one in the template library. So we can just borrow that structure, protein complex structure, as a template and build a homology model for my query sequence pair. So that's how we do it. Now, there are many other ways of doing this. Homology modeling is known to be, is the most traditional, the most conservative, and is known to be the most accurate if you're lucky. You can also do, for example, docking, you can do ab initio prediction, but these other methods are less accurate so far, but there's a lot of room for improvement for sure. So as Jake mentioned, the first demonstration of the feasibility of this approach is done by Philip Kim, Jason Liu, and myself when we were all postdocs in Mark Gerstein's lab. So we were able to show this is in yeast to show that this proof of principle that we can actually build for a subset of the nodes and edges in the binary yeast interactome network, structural models for these nodes and edges, and we call that the structural interaction network, 3D structural interaction network. And what that immediately allowed us to do is to, for example, here is to differentiate two kinds of hubs. Hubs are proteins which have a lot of interaction partners, here shown in green. Now, if you don't have any structural details, that's all you know. This protein is a hub, interacts with many interaction partners. But once you have the atomic structural details, you can tell there are actually two different kinds of hubs, right? So there's this blue hub, which interacts with all of these interaction partners using the same interface. Basically, all its interaction partners are competing for binding using the same interface. Versus this yellow hub, which binds to its interaction partners using different interfaces, right? So this is kind of a fundamental distinction between the two kinds of hubs, but you can only tell this distinction when you have atomic structural details. So I want to talk about our following research in terms of applying this structural systems biology approach, focusing on different kinds of applications. So I want to talk about its application in the disease space to both a genetic infectious disease, but as well as in the space of fundamental science, and talk about network design principles, learning about network design principles. And if I have time, I'll talk a little bit about multicellular complexity as well. So one of the really very powerful idea in network biology, in particular in relation to human disease, genetic mutations and genetic diseases, is this notion of agiotype that's proposed by Mark Waddell, Professor Mark Waddell from Dana-Farber Cancer Institute. So the idea is that you get different mutations. You can have different missense mutations on the same protein, but they're going to give you very different kinds of network perturbation patterns. And then as a result of that's called agiotypes, and it's going to give you different phenotypes. So here is an example of 4 different mutations on the same protein. So here's to the left is the wild type protein. Now this mutation, the first mutation, it lies on the surface of the hub protein, but it doesn't disrupt any interaction. So this will give you edge preservation. The entire interactome is still the same. So this is called a quasi-wild type mutation. It's basically the same as the wild type. The second mutation lies in the core of the protein. Basically, the whole protein falls apart. Therefore, all of the interactions are gone. So this is called a quasi-null mutation because it basically kills the entire protein. But some of other mutations, they have more subtle perturbation patterns on the interactome. So the third mutation here, it lies on the surface of the hub protein, but it lies at the interface mediating a specific interaction. So when you have this mutation, it's going to lead to a specific loss of a specific interaction, but all the other interactions are still there, right? So this is a very subtle perturbation of the interactome, and these are called the agetic perturbations, agetic mutations. And finally, you may also have mutations which lead to gain of interactions as opposed to loss of interactions. And each one of these different agiotypes will lead to different phenotypes. So these kinds of mutation agiotypes are very important. So Mark Waddell and other people have this entire research program in terms of experimentally mapping these agiotypes for all disease mutations. using EAS2 hybrid or other experimental screens. But you can also try to predict them computationally if you know the structure, or if you have a structural model for these proteins and protein-protein interactions. So you can do both, and they agree with each other pretty well. So I want to talk about this study, mutation agityping study by Mark Vidal. So this study is really a very large-scale study where they try to determine the specific patterns of perturbation for all Mendelian disease mutations, as well as many common mutations. So the experimental parts are led by Nidhi Sani and Song Yi in Mark Vidal's lab, and Jasmine Kuhlman-Huntington from my lab did a lot of the computational analysis. It's also co-led by Fritz Roth from the University of Toronto. So it's a lot of work, but the bottom line is you immediately see that there's a great difference. There's a big difference in terms of how disease mutations and common mutations that are non-disease perturb interactome networks. So when you look to the right pie chart, you can see that for the common mutations, those mutations that don't lead to disease, they basically don't do anything to the interactome, right? So the vast majority of them are quasi-wild type. They don't do anything to the interactome. Whereas for disease mutations, Mendelian disease-causing mutations, more than half of them disrupt the interactome one way or another. Now, what's really interesting is when you look into in detail how these mutations disrupt the interactome, half of them are the red cases. These are the quasi-null mutations. Basically, they kill the whole protein. So the whole protein falls apart. Of course, all the interactions are gone. But the other half, the blue part, are the more subtle perturbation type. So these are the agenetic mutations. The protein is still there, but the mutation lies on the surface, on the interface of the protein mediating a specific interaction. So when you have this mutation, it's just that specific interaction that's mediated by that interface. That interaction is gone. But all the other interactions are still there. The protein is still there. So most of the protein function is still preserved. It's just that specific interaction is being cut. So this turns out to be easily explained by a structural perspective. Okay, so when you map these experimentally determined agiotypes back to protein structure, so you can actually see that these so-called agenetic mutations, they're basically the mutations on the protein surface, but it's a specific part of the surface. It's inside a particular interface that mediating a particular interaction. And when you have those mutations on these interfacial residues, then it sort of perturb a specific interaction and make this mutation agenetic. Okay, so these mutations can be rationalized in terms of these kinds of structural models. Now, the reason why these agenetic mutations are important is to know which edge they disrupt is because it turns out that it's the disrupted edge that's the most important for the disease phenotype. So, for example, in this case, there's agenetic mutation. It disrupts this edge, but these two other edges, these two other protein-protein interactions are still maintained. So when you look at which one of these edges interactions are more relevant to the disease, is the disturbed one, is the one that's perturbed by this mutation, that's more relevant to the particular disease that this mutation is responsible for. So knowing which edges are specifically disrupted by the mutation is important for understanding the mechanisms of disease, genetic diseases. So this is a kind of a case study of 4 different mutations on the same on the same protein. You can see that the first 3 mutations, they have the same agiotype. What do I mean by that? Well, they disrupt the interactome in the same way. Many interactions are killed, other interactions are preserved, but it's the same set of interactions that's been disrupted for the first 3 mutations. Whereas the last mutation has a different agiotype, in particular that is just a quasi-wild type mutation. It doesn't do anything to the protein interactome. And as you can see here, that the first three mutations with the same agiotype, they also happen to have the same disease phenotype. They cause the same disease. Whereas the last mutation, even though it's on the same protein, but it has a different agiotype, and it's going to give you a different disease phenotype. So knowing these precise edge perturbation patterns give you a better predictive power for the disease phenotype. Now, all of these are done by experiments, which can be very time consuming, but you can also do mutation agiotyping using computational methods if you know the three-dimensional structure models. So basically, by using 3D structure-based predictions, you can also predict the mutation agiotypes, whether or not it's quasi-wild type or agiotic or quasi-null. And overall, the agreement with experiments is pretty good, considering the fact that these are very different technologies and they operate on a very different space of mutations, very different space of proteins and protein-protein interactions. The agreement are actually quite good. So this is the work done by Mohammed Ghadi, but I will talk more about how he does computational agiotype prediction in a bit. Yeah, so there's a question in the chat box. Is indirect relations a link between, is also influenced by agiotic mutations? Well, these agiotic mutations, they will cause a disruption of a direct interaction, but then these kinds of perturbation of direct physical interactions will propagate, right, in the space of networks, which will then affect indirect interactions. So that's the way that we would like to think about it. So we can then use these kinds of mutation agiotyping results to actually help us understand numerous network design principles. And the particular design principle that we would like to understand is, why is the human interactome so complicated, right? I mean, typically you find a map of the human interactome, so many nodes and many edges, and it's very difficult to figure out what is going on and to sort of figure out how the structure of the network affects its function. So the question is, why is the human interaction so complicated? Now, in thinking about this, there are two different ways of thinking about it, and this is showcased by this commercial. This is a commercial that's put out by Apple actually many years ago, basically to say that, well, to the left is the PC. PC is complex. Windows operating system is complex, not because it's necessary, just because it's bloated, But to the right we have Mac, which is complex, but it's lean. Everything is there for good reason. So the question is, which one is the human interactome? Is it more similar to the left or similar to the right? So you can sort of think about these two different hypotheses in the following way, right? I mean, does the human interactome network resemble a Mac? Or the other way to think about it is a Swiss watch, right? So it is very complicated, but it's because its function is very complicated. All the complexity are necessary to carry out its function. So all the pieces are useful. So this particular hypothesis is called the adaptive origin of complexity. But there's a competing hypothesis which says that, well, actually, maybe the human interactome more resembles the Windows. It's just bloated. It's complex, but it's not necessary. And the reason for why it's so complex is for a completely different reason. It's because when you're a software engineer, it's very easy to add features. It's very easy to grow your code base. But once you grow them, it's almost impossible to shrink back. So it's just it's for that reason. So this particular hypothesis is called the non-adaptive origin of complexity. So the question is, which one of these more resembles, does the human interactome more resembles? So now this is a pretty hard question to answer. So we take a particular approach. The particular approach we took is to try to estimate what fraction of the human protein-protein interactions in the interactome are so-called dispensable interactions. So what are dispensable interactions? These are real interactions, okay? These are real interactions. They're not noise. They're not errors. They're real interactions. But if you take these interactions, if you cut them, if you kill them, they're neutral. So the cell, you're still, the cell and the organism is still perfectly fine. So these are neutral upon disruption. So if you have a lot of these dispensable interactions, then you could say, well, then it's bloated and maybe resembles the Windows operating system. But if you have very little of them, then you can say that it's more like Mac or Swiss watch. So these are, so how do you figure out how much of your interactions are dispensable? Now, so here is an experiment I would like to be able to do, but unfortunately I can't. So the experiment is you take the human interactome, right? So here we have the binary human interactome, and then you take each one of these edges and you would cut it, okay? You would cut it and you would ask, does this hurt or not, right? So this particular edge, you would cut it and say, does it hurt? If it hurts, then it's a deleterious, it leads to a deleterious consequence. But if it doesn't hurt, the cell and the organism is still happy, then it's a neutral, then it's a dispensable interaction. So the question is, then you would cut these wires one by one and ask what fraction of these wires you can cut that the organism won't feel hurt. So that's the dispensable content. But unfortunately, it seems to me like a very hard experiment to do. So what we, in particular, what I mean by we is Mohammed Ghadi, who is then my PhD student, it decided to do a little bit of a workaround. So his idea is, let's try to tackle this problem from a structural systems biology approach. Okay, so let's try to first create a structural models for these protein interactions, and then map different kinds of mutations, and then try to work from there. Kind of do a mutation agile typing approach, but using structural systems biology. So he was able to build homology-based structural models for many of the proteins and protein-protein interactions in the human reference interactome. So he used two different kinds of human reference interactome. One is based on the Is2 hybrid experiments with validations, and the other one is based on other kinds of literature-based experimentally validated interactions. So once you have the human structural interactome, then you can map mutations, different kinds of mutations, onto the interactome and predict what they're going to do in terms of the agile type. And the thought process is exactly the same as I was telling you before. If you have a mutation that's lying in the core of the protein, and if it disrupts the overall stability of the protein, the whole protein will fall apart, then you're going to predict that mutation is a quasi-null mutation. But if you have a mutation that lies on the surface of the protein, but it lies on the interface, and if that mutation actually causes there to be a significant change in the binding free energy, then you will predict that it's going to be an agenetic mutation. Otherwise, if it lies on the surface of the protein, but elsewhere from the interface, then you're going to predict that it's a quasi-wild type mutation. So you can use structure-based and physics-based calculations to predict the agile types for different kinds of mutations. So what Mohammed did is to look at different kinds of mutations, both the pathogenic mutations, the Mendelian disease-causing mutations from clean VAR, as well as common non-disease-causing mutations from dbSNP. And then he wants to compare how these two kinds of mutations affect the interactome. So this is the results that he got from his structure-based calculations. You can see that they agree very well with the experimental agile typing results from Sunny and co-workers. To the left, you can see that most of pretty much all of the common mutations, they don't do anything to the interactome. All right, so pretty much mutations are quasi-wild type, but there are a few exceptions. A few of them do disrupt the interactome. Whereas for disease mutations, Mendelian disease-causing mutations, a significant fraction of them agenetically disrupt the interactome. So they happen at the protein-protein binding interface, and then they would disrupt those specific interactions mediated by those specific interfaces. So, and when you look at these two pie charts, and you realize that, well, if you assume, for example, that common mutations are neutral, because they don't, we all have it, and they're all okay. We're all okay with them, but Mendelian disease-causing mutations are deleterious because they're harmful. Then from these pie charts, we actually know the conditional probability for a neutral mutation versus a deleterious mutation being agetic. The probability that the neutral mutation agetic disrupts a specific protein-protein interaction edge, or the deleterious mutation, the probability for deleterious mutation to specifically disrupts a protein-protein interaction edge. So we know the probability of agetic given that is neutral. But what we really want is the dispensable content, which is the probability that each edge, when we disrupt it, is going to give you a neutral effect. It won't cause any pain. So it's the other way around. It's the probability of neutral given agetic, you see? So we have to find a way to switch the two sides of the conditional probability, and we have a way of doing that. That's called the Bayes rule. But we do need to know a few other things. We need to know the prior probability for these mutations to be neutral or deleterious. We need to also have this normalizing constant on the bottom, which is easy to get rid of. But once we have all of that, we actually can flip the two sides of the conditional probability, actually calculate what we really want is the probability of neutral given its agetic mutation. And this is really the dispensable content. So make a long story short, we're able to estimate what fraction of the human interactome are neutral upon disruption. And the answer is a fairly small fraction. It's certainly the most of the interactome edges, when you kill them, when you cut them, is going to be deleterious. But there are less than 20% of these interaction edges, when you kill them, they're actually okay, they're neutral, so they're dispensable. And this agrees pretty well between the computational predictions based on structural systems biology calculations and experiments from mutation agiotyping experiments. So I think I'm going to skip this. And I want to switch gears and talk a little bit about infectious disease. So this is another domain of application for this structural systems biology approach. So of course, this has received increased attention in recent years. So there has been a lot more data coming out in recent years. But the study I'm going to talk about is actually the first study in this space, which we did a while ago, back when we have relatively little, still a lot, but today we have a lot more data. So this is work done by Eric Franzosa, but then PhD student with me. So he first built a structural models for the human interactome, the protein-protein interactions in human, but then he thought, that, hey, there's actually a whole different kind of interactions, right? The interactions between different species, in this case, between the human and pathogen, which people have never really been thinking about, at least in the space of structural system biology. So he collected all the human pathogen interactions that he can and build structural models for them. It turns out that most of them are human virus protein-protein interactions, for obvious reasons. So he was able to build this structural network for not only for human proteins, but also for virus proteins, and not only for human protein-protein interactions, also for human virus protein-protein interactions. So this is a very interesting network because it has two different kinds of edges, right? Different kinds of nodes and two different kinds of edges. There's the endogenous interactions between two human proteins, as well as exogenous interactions between the human protein and the virus protein. So the network he built back then was pretty small. So here's the network. So each one of these red dot is a virus protein. Each one of these red lines is the interaction between the viral protein and the human protein. Everything else are human proteins and their endogenous interactions. But still, this is a structural network, which means that we have structural models for all these nodes and edges in this network. Now, when we have this network, there are some obvious things we can do. The first obvious thing we can do is we can investigate what is the geometric relationship between different edges. If, let's say here, we have a case of a viral protein and a human protein, they target the same human protein, but these are two different edges. So the question is, how do they relate to each other in terms of geometry, in terms of structure? Do they have nothing to do with each other? So that's the first possibility. Or do they tend to overlap with each other in terms of these red and blue interfaces, right? These exogenous and endogenous interfaces. So that is an obvious question you can do, which we will not be able to do if all you have is the binary network information. So we really need the structural information in order to know the answer to this particular question. So to cut a long story short, what Eric was able to find is that it turns out that second scenario is the correct scenario. That whenever a virus and a human protein binds to a third human target protein, there's a significant tendency for these two interfaces, the exogenous interface in red and the endogenous interface in blue, to significantly overlap with each other. So what this means is that there's this notion of, this underlies this notion of mimicry, right? Viral mimicry of human, but it's a particular kind of mimicry. It's an interface mimicry. Is the viral protein and human protein, they mimic, the virus protein is trying to mimic the human protein in terms of the interface of binding. Now you can ask then, how does the virus achieve interfacial mimicry? Now there are two possibilities. So on the top is the first possibility. So here we have a viral protein in red, mimics this human protein in blue. They compete for binding to the third human target protein in gray. But in this case, you can see that the mechanism for interface mimicry is that the virus protein looks exactly the same as the human protein. It's basically a copy of the human protein, right? So that's an easy way of achieving interfacial mimicry, is by sequence and structural similarity. But at the bottom, you see another case in which here's a viral protein in red, mimics a human protein in red. They bind at the same site of the third target protein, human protein in gray, but here the viral protein looks completely different from the human protein. So this is interfacial mimicry without any sequence and structural similarity. So you can then ask, how often does the first scenario happen, and how often does the second scenario happen? And it turns out that in most of the cases where a virus mimics the human in terms of interfacial mimicry, the virus protein looks completely different from the human protein. So most of the cases are achieved by the second scenario. But when you look back into the human interactome itself, because there are a lot of cases of human protein mimicking other human proteins, okay? And when you look at those cases, it turns out that most of the within human interfacial mimicry are achieved by essentially copy and pasting. The human protein, the two human proteins look very similar to each other in terms of sequence and structure. So this is a big difference between the mechanisms of interface mimicry between the virus-host interactions versus the within-host interactions.
So Brendan, can I ask a quick question?
Yeah, sure.
Yeah. So even for the first case, I thought evolutionary, it would be almost impossible to evolve the same sequence and the structure for the virus. How does it happen even? I thought that it's basically zero.
Yeah, that's a really good question. I think that what happens is the viral genome just steals a protein from the host. So, or vice versa. It's basically horizontal gene transfer. Or vice versa. But most likely, I think it's a case of it's not a case of convergent evolution. Yes, you're absolutely right. There's no way you can convergently evolve another protein with similar. You can convergently evolve a protein with similar structure, but there's no way you can convergently evolve a protein with similar sequence. Yeah, so these are most likely cases of horizontal gene transfers. Yeah, that is a very good question. Yes. So in any case, and then you can ask the question, okay, but the human interactome is huge, right? There are so many interactions, so many binding sites, so many interfaces. Why do the viruses only target these interfaces, but not so many other interfaces? So you can do a bit of a data mining bioinformatics and ask, are there any differences between the interfaces that are mimicked and targeted by the virus versus all these other endogenous interfaces? And it turns out that there is actually quite a bit of different. They're really different. So these mimic interfaces targeted by the virus, they tend to involve regulatory activities in the human. They tend to involve transient binding, and they tend to mediate many endogenous interactions. Basically, these are, you can think of them as important, high-level important interfaces. And the idea here is that virus genomes are tiny. They have very limited genomic resources, so they really cannot target everything they want to target. They have to pick and choose. You really want to target interfaces, which will give you a big sort of consequence. So this is kind of parts of the evolution pressure that's putting on these viruses. Then the last question you can ask is, okay, you have all these different interfaces. You got the endogenous interface that's binding to human proteins. You got the exogenous interface binding to virus proteins, and they also overlap. You have this purple mimic interface that's targeted by both human and viral proteins. So are there any other things that makes them different? And it turns out that yes, their rate of evolution is very different. Their divergence, their sequence divergence, level of sequence divergence is very different. In particular, the blue part, which is the endogenous interface, they are the most conserved, which makes sense, right? You got a protein complex, the interface is super important. If you have a mutation there, most likely it's going to destroy the whole machine, so you don't want any mutations on your interface. So the endogenous interface is very conserved. But what's interesting, turns out that the purple part, the middle mimic part, turns out to be the most divergent. It diverges the fastest, in this case, between human and mouse. So this underlies this notion of evolution arms race between a host and pathogen, right? So the virus is trying to target the human protein, and the human protein is trying to run away from the viral protein by accumulating these mutations at the mimicked interfaces. So this is a demonstration of that. So the other interesting thing you can also learn from this is from this sort of a structural systems biology perspective, is this notion of viruses as perturbagens of networks, just like genetic mutations as perturbagens of networks. So this is the work done by Frank Chen, then a PhD student in my lab. So what he was able to show is when you have a virus targeting an interface, and you also have a mutation targeting the same interface, even though it's a very different cause, it's a very different perturbation, but as long as they target the same interface, they will give you similar network perturbation patterns, and in the end, they actually give you similar disease phenotypes. So in a way, you can think of the virus as some kind of a network perturbagen, and it'll give you similar disease phenotypes as similar to genetic mutations in this regard. So I guess I have a little bit of a time left. I'll briefly mention the last part of this structural systems biology application is to probe what makes multicellular organisms, what gives this additional complexity to multicellular organisms. And I'm going to focus on alternative splicing. So many of you know, we have 20,000 genes in the genome, but we got millions of proteoforms, right? And the reason for that, one important reason for that is there's this mechanism called alternative splicing, which will generate multiple isoforms based on the same gene, based on the same gene from the genome. So the question is, these multiple protein isoforms generated by alternative splicing, are they all pretty much the same thing, or are they totally different from each other from the perspective of network biology? So that's the kind of question. So this is, again, a collaboration with Mark Vidal at Dana-Farber Cancer Institute and Lydia Iokachova at University of California, San Diego. So the experimental part led by Xinping Yang from Vidal lab, and the computational part led by Jasmine Kuhlman-Huntington from my lab, and Shuli Kang from Lydia's lab. So the idea is to take all the protein isoforms, not all, but many protein isoforms in human, and then map their interactions with other human proteins, and basically ask, do different isoforms of the same gene, do they interact with the same protein partners or not? So this is called the isoform resolved interactome network. So it turns out that many of them do. So here are the identical cases. Many of the isoforms which are generated by the same gene, they do interact with the same set of proteins, but many also don't. So on the top, you can see the distinct, to the extreme, you actually have distinct cases where the isoforms of the same gene actually interacts with completely different protein partners, which is a very interesting case. And there are many of them. So this phenomenon is actually quite widespread. So this turns out to have important ramifications for both in terms of basic biology and in terms of human disease. So you can see here, you can look at for pairs of proteins, pairs of isoforms that either that either bind to the same set of protein partners or a completely different set of protein partners, they have a significant difference in terms of their expression, co-expression in different tissues, as well as in terms of their disease outcome. So this is something which is important if we wish to sort of study the consequences of isoforms in terms of tissue expression and disease phenotype. So when you look deeper, it turns out that many of these kind of remodeling or rewiring by alternative splicing can also be explained in terms of protein structure, in terms of structural systems biology. So it turns out that you find a lot of different cases, roughly equally likely. So you find a lot of cases where you got a region of the sequence in which if you splice it out, then it leads to a loss of interaction. So that region is considered an interaction promoting region, right? If you cut it, then you lose that interaction. If you put it back, you gain that interaction. So what does that mean? Well, there is a domain there which mediates the interaction. So if you cut that domain out by alternative splicing, you lose that interaction. If you put that domain back in, you gain that interaction. There are many other complicated cases, unfortunately. So for example, there are interaction inhibiting regions. So when you cut it out, you actually gain an interaction. And when you put it back in, you lose an interaction. So why is that? And so you can think of different reasons for that. And there are also complex cases, but fortunately, the complex cases appear fewer than we'd expect by chance. So most cases are explained by these single regions of the gene, which you cut it out or you put it back in, it leads to either gain or loss of a specific interaction. So, so because of that, we can actually use structural systems biology predictions to predict how isoforms are going to perturb the interactome. This is the work done by Mohammed Ghadi. And what we try to argue is that these predictions, they perfectly complement the experiments because they probe different parts of the protein space, of the human genes, of the human gene space. So they very nicely complement each other. So he was able to build, so the way he predicts, it's actually fairly straightforward. So here's an example. Here's a gene that encodes 3 domains. Now, 2 of these are interacting domains. So LB domain interacts with EGF, whereas PK domain interacts with GRB2. So the prediction goes, okay, let's say this is the reference gene, but if you have different isoforms of the same gene, depends on which domain that has been cut out from the reference gene, then we can predict which interactions has been lost. Okay, so if we cut out the PK domain, then we can predict, okay, so GRB2, this interaction has been lost. Whereas if we cut out some other domain, then we can predict that those other interactions mediated by those interactions, mediated by those domains has been lost. So basically, this is how you make these kinds of predictions. And Mohammed was able to show that these predictions are generally of high quality when you cross-references it with function annotations, with co-expression patterns, tissue co-expression patterns, and whatnot. So the bottom line from this work is just like in the experiments from these computational studies, we confirmed that there is actually extensive network remodeling by alternative splicing. Roughly more than 20% of genes with numerous isoforms have at least one isoform losing an interaction. So there's quite a bit of interaction rewiring events, and we really have to think about them. We really have to think beyond just one gene coding one protein in the interactome, but really have to look at these isoforms in more detail. So I want to close by summarizing that I hope to have convinced you that this structural systems biology approach holds a lot of water, is something to worth thinking about, and it gives you insights into a variety of topics, including basic science, network design principles, but also in the disease space, genetic and infectious disease. So I think I ran out of time, so I want to thank you very much, and I'll be happy to take any questions.
Yeah, thanks. You're okay with the
All right.
time. We do have a few questions in the chat. So thanks for the great presentation. The first question is indirect relationships. Are they also influenced by genetic mutations? I think you just talked about that, but this question came early on.
Yeah, but I think it's a great question because the way to think about it, I think, of course, I focus on physical interactions in the interactome, right? The physical interactions, direct physical interactions. But there are many very important indirect interactions, indirect relations, going from genotypes to phenotypes. Those are indirect, very important. The focus of genetics and medical genetics is this relation between genotypes and phenotypes. But I think that those should be interpreted in terms of a cascade of physical interactions, a direct physical interaction. So that's how I would like to think about it. Yeah.
Okay, next question. Is it necessary to follow power law to assume these hypotheses?
No, no, it is not necessary. Yeah, it's not. It works for any networks of any topology. But the important thing is you have to start with an experimentally determined network interactome. So we didn't predict any new protein interactions.
Okay, next question. Are the interactions between protein or human virus,
yes,
are they experimentally validated?
That is a very good question. The same as before. So we start from experimentally determined protein and human and human virus protein interactions. So again, we were not trying to predict any new interactions between these proteins. What we do is we take experimentally determined protein interactions, but then try to add atomic details, try to add structural details, atomic details by annotating these protein interactions.
Yeah. If mutation enhances instead of disrupts protein interaction, would there also be a way to systematically predict such gain of function mutations?
That is a very good question. I don't really have any sort of useful insights in that. I think gain of, you can see that I'm basically talking about mutations disrupting protein interactions. Why? Because those are easy to predict. You have an interaction, you already have an interaction, then you have a mutation on the interface, and then it's easy to predict which mutation disrupts the interaction. How do you deal with gain of function or gain of interaction mutations? Honestly, I do not know, but I think this is an area which certainly worth further exploration, because gain of interaction, gain of function mutations are, as you know, very important.
Yeah, well, this is, there's just so many complicated ways that these things can manifest. So, right, you have gain of function, you have excess of certain substances, or sometimes you have irreversible binding when it binds and then nobody else can bind to it. So there's lots of different ways that could work.
Yeah.
Another question, you use presently available tools like SIFT, Probian, or CAD scores? And if yes, how do you set the score threshold?
So we don't use these tools. If I understand correctly, SIFT is a tool which allow you to predict the severity, essentially predict the disease phenotype, whether or not a mutation is likely to be a disease mutation or not.
That was another question that I skipped. Sorry. What about the dynamic aspects of network interactions? You're describing a flat network.
That is also a very good question. Yeah, it's a static net. It's a static network. So, but the way I would think about that is, first of all, you got to start from somewhere. Having a network is better than not having a network. But then once you have the network, what you can do is you can map dynamic properties onto the network, right? So you can do rich annotations of the network. You can map, for example, gene expression dynamics or tissue expression dynamics onto the network, and then use that to tease out the dynamic aspects of the networks. But you're absolutely right. This is really just a starting point, and there are many other, very exciting avenues that one can explore. Yes.
Other questions? We still have a couple minutes before we reach the final end.
Yeah, I'll ask a question. Yeah. Hi, Brendan. Yeah.
Hi.
Can you hear me? Yeah.
Yeah.
Yeah. So great work. You mentioned about the protein isoform, and we also studied that before. I know there are many computational efforts to predict the isoform interaction through machine learning, deep learning approach. but now you know that the AlphaFold 2 have predicted hundreds of millions of proteins, structures, including all the isoform. Are you or do you know of groups that are systematically looking at the binding energies of normal and isoform-based structures, and then actually to solve this problem once and for all, and let's say augment the happy database that we have to be isoform specific happy?
Yeah, I think that is certainly something that'd be a great direction to go into. I'm not aware of any kind of systematic efforts, but I will say that even just in this case of how isoforms disrupt protein-protein interactions, that by itself is actually there are many different scenarios, right? So you could have, so I was saying you could have an isoform which you splice in an interacting domain, then that interacting domain will then brings in all these interaction partners, right? So these are or you can slice that out, and then you will lose those interactions. But you also have exactly the opposite phenomena, where you splice in the region, and then you lose a bunch of interactions. You splice it out, you gain a bunch of interactions. So why is that, right? So again, it's one possible justification for that is you may have, when you bring in that interaction domain, actually interacts intramolecularly with another interaction domain in the same protein. So it's basically kind of like an inhibitor, to another interaction domain of the same protein. So if you cut that inhibitor out, then you free the other interacting domain so that it's free to interacting whatever is trying to interact. But what I'm trying to say is, so those are certainly cases which one can predict. So one can predict using either structure-based calculations like AlphaFold, or using the data-driven machine learning approaches, deep learning approaches. Part of the challenge is the lack of training data, I think, is just that there are just really, the kinds of isoform-specific interaction data they do exist, but they're not a lot, right? So it's really, in many ways, in this aspect, it's big data for protein sequence and protein structure, but it's seems to me it's still in a little bit smaller data era for the isoform-specific molecular interactions. But I think that this is certainly a space in which I'm not aware of any. I think this is a great direction to go into.
Yeah. And another very quick before we close out, since you have so much knowledge about the protein interactions, can you actually find a potential drug target and then critical interaction partners, and carve out stored peptides to design peptide drugs? Do you know of people who are doing that? Or since you're in biomedical engineering department, maybe you're closer to some kind of a peptide therapeutic based on this?
Yeah, that's also a really great question. this whole idea of targeting edges, right, as opposed to nodes for therapeutics is I think it's a very exciting area. I'm aware of a few. I think the Toronto U of T, there are a few people who's working in that space. But I think this is an area which I think it holds. I agree with you. It holds really a lot of promise, because that's where you want to target. You want to be able to target specific interactions that mediates a particular disease phenotype. I think that is where people want to go. Yeah, but
and there's another question in the chat. What about protein modification in a signaling network?
Well, those will give you even more proteoforms. So as if the millions of alternative splicing mediated proteoforms are not big enough, then yes, you're going to have even more proteoforms, more protein species in the human cell that we have to worry about. Do they affect protein-protein interactions? Of course. So you're going to even have even more complexity to deal with. Yeah, but those can be measured, I think, quite readily by mass spectrometry these days. So you can try to overlay these kinds of information on top of the interactome, and then be able to tease out even more information.
Yeah, and can you hear me, Brandon?
Yeah.
Yeah, well, thank you. Great talk. I was the one who asked about the dynamics and also the protein. So proteins can be moved in the cell as part of signaling. So when you're looking at an interactome, you have to build into the possibility that do the proteins actually ever come together? Because
Yes.
the cell is not a homogeneous environment. So you've got a partition. I was thinking of an analogy when you were talking, and maybe this is a little bit of a Schrodinger cat scenario. So think of a closed room. You're in it, and in the room there is a bucket of potassium cyanide, and it's linked to a little bit of acid. So if you run into it, you'll die. So what are the chances that you will interact with it? So does chance play a role here? I mean, we tend to think that protein-protein interactions are deterministic, but they're actually not. Basically, proteins bump into each other randomly, and then because of their interaction, they stay together for a period of time before falling apart again. The signaling network is built, as I said, very, very flatly. How do you build that into your model?
That is a very, that's such a brilliant question. So I completely agree with you. I mean, there are so many different, not all protein-protein interactions are the same, right? So there are just so many different types with different flavors, So you have like you said, proteins bumping each other all the time. So that's sort of stochasticity, intracellular stochasticity play a big role in that. But then there's also the transient interactions, right? The two proteins, signaling proteins, they come together, do something, and then falls apart and do something else. Then there's the permanent interaction, like molecular machines, the ribosomes and the proteasomes, all the parts have to be assembled before the whole thing can function. And then lately, there's also all these talk about condensates, right? Biomolecular condensates, those are pretty non-specific, but they're quite important. So how do you think of all of these interactions? I think that that's part of the challenge, is how to put them into this kind of a really very highly abstract graph theoretic framework, kind of put into it a lot of the biological interpretation. I think that really is the challenge. So I really think what we are trying to do here is just the first step. You got to put in the atomic details, but that's far from, you got to put in all these dynamics and these localization issues, and also think about sort of the consequence, the functional consequence of these different encounterings. Yeah.